Thanks for joining us here at The Research Her, the show working to improve the health disparities for women of color, one topic at a time. And I am Alicia. I'm here learning and growing with you as we research our way to wellness. Get into this episode, okay? Because our Graces breaks down viruses for us, teaches us about antibodies and vaccines. We are here to learn. We get into central dogma and all type of things. Who are we talking to today? Our great sis, Natasha Duggan, who was born in Bulawayo, Zimbabwe, and immigrated to the United States at the age of 3 with her mother. She earned a Bachelor of Arts in Psychology from the University of San Francisco. Following college, she spent some time, about 3 years, working in a laboratory at the University of Virginia, studying angiogenesis in diabetic individuals. She decided to continue her education, so she moved to New Orleans and attended Tulane University School of Public Health, where she received her Master of Science in Public Health with a focus in tropical medicine. And we're not done yet, okay? From there, she went on to the University of Miami, where she is currently completing her PhD in cell developmental biology. Her work focuses on isolating, neutralizing antibodies against HIV and SIV as potential tools for HIV vaccine development. So let's get into this conversation with our great sis, Natasha. Welcome, Natasha.
Thank you for having me.
Let's just get started. Our great sis has been in all of the institutions, studying all of the things, okay? So we got some biochemistry lab experience. We have some psychology, masters of public health. We're working on our PhD in cell developmental biology. Talk to us about that and how did they all end up coming together to get you exactly where you are today?
Education-wise, I started out as a physics major. At the University of San Francisco, they said the average graduation time for physics majors was 6.5 years. I just couldn't do it, but I always thought the mind was interesting. So I got my psych degree. After college, I went and I worked, got real life experience in a lab, which is where I really found I had a love for microbiology and infectious diseases. And I also realized I could not be at the bottom of the food chain when it comes to having a job. So I knew I needed to get more educated so I could be the person in the lab making the decisions. Also, I've always had this interest in public health, and the program at Tulane, they have a Master's of Science in Public Health with the focus of tropical medicine, and the only other program like that in the world is the London School of Hygiene. So I knew I was going to get this degree. And to me, the question was whether or not I was going to get it before or after. I went for a PhD, and I was advised that after I did a PhD, I would probably not want to continue my education. So I decided to get the Master's of Public Health before. And there you do your basic public health requirements, but you take like immunology and tropical virology and courses like that. So that was exciting. And I also got to work in a lab doing virus work for the first time. So that was good. And then I finally got to the point to do my PhD. And I came to the University of Miami, and I chose here because they had a lot of HIV researchers. When I wanted to become a scientist, I knew that I wanted to make an impact, which to me meant I had to work on something that affects lots of people. So from a public health perspective, that meant I was either going to be working on malaria, tuberculosis, or HIV, because those are the big three when it comes to public health concerns. And TB honestly still scares me. The way it's transmitted is through the air, and it made me anxious to actually work with it. But also being born in Zimbabwe, in Southern Africa, and seeing how HIV ravaged that country, even if I'm only doing like the research side of HIV research, I feel like I need to do something to help because it's heartbreaking to see what HIV has done to it.
It's interesting, though, the path that you took. Specifically, I'm intrigued at the Master's of Science in Public Health with the focus on tropical medicine. How did you learn about that program?
My mother had always talked about the London School of Hygiene. She always tried to encourage me to go get educated places that weren't always the United States. I wasn't necessarily down for that, but I found Tulane had a program that was basically the same, because really I like infectious diseases, and the things that are tropical diseases are really the ones that impact most of the world. I feel like in the US, so much research is focused on things that affect Americans, but Africa, Asia, they matter, and the people there matter. So I think those diseases are really important because those are a lot easier to deal with than your diet is causing high blood pressures or diabetes. I think those are a lot bigger things to tackle. So I looked at the program and I saw all the science courses in the virology.
Why did you choose to study public health as opposed to continuing on the lab benchtop work?
What I believed before, and honestly, I believe it more now in this current pandemic, is I feel that scientists and public health professionals do not have a good marriage when it comes to working on these diseases that are impacting so many people. And to really be able to make an impact on things, you need to understand the biological science of it, but you also need to have public health application, or else you're only doing half the work. And it's become so evident that those people need to exist now that are able to be translational in this pandemic, because I feel like a lot of people who are making the decisions or the people who are communicating to the communities are scientists, and they're not breaking it down. They don't have the education or the skill set to really be able to reach the public the way that they should.
What has been the greatest research experience that you've had thus far, not just as a PhD student, but even possibly before that?
I think the most exciting thing that happens is the first time that you're like an author on a paper. For me, that was like a moment where I was super excited. I got copies of the paper. It wasn't like I wasn't first author, but I was like in my early 20s, and I was like, the work I did has been written up and now it's being shared with people. And I'm like, I wrote these paragraphs and I was part of that. And I think that is why you do what you do. You find out the new things so that you can tell other people about the new things.
Have you had any challenges that you're willing to share with us?
I don't think a PhD is the easiest time in anyone's life. I think a PhD forces you to really light a fire under yourself and push a lot. I don't think academia always has just the healthiest relationships with their students. So I think going through a PhD is also a mental Olympics, along with the fact that it takes a long time and it has no end date. So that's a bit challenging, but I think if you know what you're getting into before you do it, that's helpful. It's also being a Black woman in this field that's dominated by white men. You feel othered sometimes, but you also feel like you need to work so much harder, and there aren't necessarily people around you who have had the same experience as you. In that aspect, I felt like I was blessed to have my mother. She also has a PhD. So it was kind of seeing a Black woman and what success can look like, and that there's an end to the tunnel and there's another side where things are pretty.
Can you tell us about your research?
Currently, what I'm doing for my thesis project is I am isolating antibodies from a monkey that is infected with SHIV. SHIV is simian human immunodeficiency virus. Naturally, monkeys are infected with SIV, which is simian immunodeficiency virus, and humans are infected with HIV. And the core of the virus is SIV, so it's capable of replicating in monkeys, but the outer coat of it has the HIV coat. So you're capable of studying human antibodies, but in a monkey model.
What is the overall goal of isolating one of these antibodies? How can this translate into the bigger picture?
Neutralizing antibodies are very difficult to isolate. If I have a neutralizing antibody, this story would be, we have this neutralizing antibody that was isolated from a monkey. We used it in an infected animal, and the antibody was capable of neutralizing the virus in the animal, and we didn't have the immune system of the animal having any issues with this treatment. That would be one avenue. That story could also go the other way and be like, we isolated this and we're still having an issue with the immune system not recognizing it, which means that the issue is not with the fact that the antibodies that they've been using are from a different species. The other avenue for another paper would be we isolated antibodies from a HIV-infected monkey. And this is what they look like. This is what they bind. This is where they bind the viral envelope, because that hasn't been shown yet.
What is an antibody?
An antibody is a protein that is created by your body in response to something foreign in your body. For example, you get a cold. Your body notices it's not supposed to be there, and your body's immune system will make antibodies that essentially will bind that virus and prevent it from being able to enter your cells and infect more cells and make you sick.
What happens if your body doesn't produce those antibodies?
A few types of immunity. So you're not completely in trouble, but if your body is not capable of producing antibodies, you are an immunocompromised person. And essentially, when a cold gets into you, your body is not going to have all the tools it needs to fight it off. Antibodies aren't the only method of attacking things. So your body probably will be able to clear your infection, but it will take a lot longer. Antibodies come from B cells. We also have T cells, and your T cells would still work even if you don't make antibodies.
If our body produces antibodies, what is the purpose of vaccines?
First of all, I wouldn't recommend just getting viruses. Some of them are easier for our bodies to fight off, like the common cold. I mean, even we can use what's going on right now, coronavirus. Some people's bodies fight it off very easily. Other people end up being intubated because their bodies aren't fighting it off appropriately. If you're vaccinated, you don't actually have to go through the pain of an actual illness to have the antibody response. The vaccine exposes your body safely to whatever is going to make you sick, and then your immune system is ready so that the next time it sees it, or in the case of a vaccine, the first time you see this disease actually come to your body is ready to go to fight off whatever infection. Because the process of making antibodies and having a strong immune response really takes 2 to 3 weeks. So if you've already seen it, that's no longer 2 to 3 weeks. That's 2 to 3 days.
What do you mean safely?
Vaccines, there are different types of vaccines. They have like killed virus vaccines, which they really just take the whole virus and they kill it and put that into your body. They have live attenuated, where they take live virus, but they take out the parts that would really let the virus get into the cell and make you sick. Or more commonly, what's done now is they take the piece of the virus or a pathogen that is really necessary for replication, the thing that your immune system is really going to target, and they just put that small piece in your body. And then your body has an immune reaction to these pieces of protein that you don't actually have to get infected with the virus for your body to be like, oh, I've seen that protein, even though now it's on the whole virus. So it attacks the whole virus if you get infected.
Is there a reason why your body still responds to a killed virus when it's introduced to your body?
It still has sequences and proteins that are super foreign to your body. A live virus or just the small piece of a protein, I think, is better. The problem with live attenuated viruses is that sometimes the virus hasn't been attenuated enough, and you might actually get the illness that the vaccine is trying to prevent you from.
What are some ways that viruses can enter the body?
Different viruses target different cells that they want to infect. A virus doesn't just enter your body and pick whichever cell it feels like and start causing havoc. Like with coronavirus, it attacks your lungs. And it does that because the receptor that it binds inside the body, once corona gets into the body, in order to enter your cells, it needs to bind a receptor called the ACE2. And that is located in the cells that are located in your respiratory tract. If you look at a virus like HIV, which is bloodborne, that virus will enter your body anytime that essentially your body's open, like a cut or like mucosa, like in your mouth. If that's open, the virus will enter there. And then from there, go into your bloodstream and then target the immune cells, which are its goal. There are also viruses that are transmitted by the fecal oral route of transmission, which typically happens when people don't use their hands after using the restroom. And those viruses are different because they're strong and tough enough to survive the stomach acids before they're capable of infecting you. An example of that is polio or hepatitis A. And then some viruses, all they need to infect you is just skin-to-skin contact. And that's the case with the herpes viruses and human papillomaviruses.
Now that we're talking about it, hypothetical situation. Really wonder if you have on a mask and you're having unprotected sex, though, if the coronavirus was to enter into the body via the reproductive system, would it then go ahead and disperse throughout the body?
As of now, what they know of corona, it cannot be sexually transmitted, but you also never know what they're going to find out, because Ebola is a disease that they thought could only be transmitted through the blood, not necessarily all your bodily fluids. But then they found out that it could also be transmitted through sexual intercourse, and that the virus likes to stay in the testicles for months after infection.
What happens when viruses enter the body?
I guess HIV is an example because it's the virus I know the best. So when HIV gets into your body, the likelihood of infection when you have an HIV exposure is not very high, but since there's 100% case fatality with HIV, it's scary. But the virus goes into the body, it's in the bloodstream, it's looking for the cell that it targets, which is the CD4 T cell. The virus has proteins on its surface that bind with only certain cell receptors that allow the virion and your CD4 T cell to fuse together. And then once they're fused, the viral particle releases the RNA into your cell. And then once the RNA is in the cell, it goes to the nucleus, makes DNA, it integrates into the host genome. Viruses don't come with all the tools they need to replicate. Viruses need a host to survive. That's part of what makes them viruses. They come with like the bare minimum they need to replicate, and then they use the host cell to make more and more viral particles. And with HIV, either these cells are released, leaving the cell intact, but that's more virus now to infect more cells, or they make the cell explode, releasing the virions also. And then they keep targeting the same cell. That's why usually with HIV, you probably have like, if it went untreated, it's 10 years where your body's trying to fight it, but it's also fighting it. And after 10 years is usually when your body starts to lose the battle.
Lately, we've been talking a lot in the news about virus mutation. What is virus mutation?
Virus mutation happens when essentially something changes in the process of copying. I was just talking about what viruses do is they use the host to make more copies of the virus. They're trying to do it rapidly. The goal of all viruses really is to make as many viruses as they can. And if it's a good virus, try not to kill the host in the process so that the host can get other people sick. A virus just wants to spread everywhere, especially with RNA viruses, which corona is. When you're making these copies of the viral genome, mistakes happen sometimes. You're doing it quick. In your body, when that happens with a cell, there are proteins that go back and fix the mistake. They're like, you're not supposed to look like this, you're supposed to look like this. And they fix that mistake. So then maybe virus A that you were infected with doesn't look like virus B that was made. And the differences in that are a mutation. Sometimes mutations do nothing to a virus, but sometimes they change the fitness of the virus. And when I say fitness, I mean how capable the virus is of avoiding your immune system, surviving outside of the body. With coronavirus, the mutations that we've seen happen so far, they haven't been that many mutations, but they've made the virus more contagious because the original coronavirus did most of its replication in the lungs. And then there was a mutation, and that mutation made the virus highly replicative in the nose and throat. And that made the virus more fit, and it was able to spread quicker. So then that became the dominant strain that we saw because it was just better at doing what viruses are trying to do.
The vaccine is out for coronavirus, and a lot of people are on the fence. I definitely understand. But one of the questions that I see constantly being asked is, how effective would a vaccine be if the virus has mutated?
So with the vaccines that are out now, that have been approved, that's the Moderna and the Pfizer, they are RNA viruses, but the protein that's going to be made from the vaccine, it's a spike on the surface of the virus, and that spike is what allows the virus to bind to the human cells. So all that's being made is this tiny little protein. The problem with the mutations that are occurring, some of them may occur somewhere else on the virus where it's not a big deal. The ones that we're really paying attention to in the media and science right now are any mutations that happen on that spike, because then that means what we're priming our immune system for is not the same as what we're going to see. The mutations that are happening are very small, like single point mutations or mutations. It's not the entire protein that is being changed. So getting a vaccine is still worth it, even if the virus is mutating, because even if it's not exactly what you want, it's still going to give you some protection. It's like with the flu vaccine. If you get it 1 year, you'll be like completely protected that year, but the flu changes year to year. The next year, if for some reason you don't get your flu vaccine, you may get the flu, but it's going to be nowhere near as bad as it would have been if you hadn't been vaccinated at all. The South African variant, they're conducting studies, have found that people who have been vaccinated, as well as people who had a very strong immune response from a natural infection, are still capable of neutralizing the new virus strains with the current vaccines and things that we have.
What you're saying is the virus is going to enter the cell. It has this code that then our cells would actually read the code and do exactly what this RNA is telling it to do, which is produce more of these viral proteins.
They can integrate into the host because what's inside our cells is the DNA. DNA gets made into RNA, RNA makes proteins. That's the process. And since that's what our cells are doing all the time, the system for checking that the DNA is being duplicated properly is very good, because that's what our body is doing to make sure that the cells that we're making have the same genetic code as the cell next to it. But our cells don't start at this RNA level. So what is actually checking if the sequence is right, the virus brings it in with it. And it's not as thorough. It's more okay with mistakes.
What is it like for you to examine antibodies?
What I find cool about my project, I mean, I guess it's less cool. It was cooler like 5 years ago, but science keeps moving. Science keeps moving. But I just think if you think about how small a cell is and all the information that's inside a cell, what I'm doing is I'm isolating antibodies from single B cells. From like a tiny cell, I'm getting the genetic information out and then making essentially what that cell would have made, but in copious amounts so that I can test it. So I take the DNA from that single cell and I amplify it. And then I take essentially that amplified product and I put it into, it's an expression that allows for high levels of replication of this DNA, because in order to actually make copious amounts of antibody, you need a lot of DNA. And then after I get this DNA, I put them into cells that essentially will spit out an antibody. If I put the DNA from the parts of antibody in it, which is what I've made and isolated from the cells, then I incubate it for a week or so. And after a week, I have a flask full of antibodies that need to be purified. And once I get my antibodies, I test them essentially to see where they're binding and if they neutralize virus. And I test them against various strains of HIV. And I also look at where they are binding on the viral particle.
How many antibodies have you looked at over the course of your training?
I've isolated probably about 200. The technology now, you can do so much more because what I'm doing is I'm doing the process of making the whole antibody, and then I'm testing all these things about it and characterizing it. The newer technology, now what they do is they essentially take those individual B cells that I was talking about before, and they make those produce lots of antibodies. And then they test the media that those antibodies are in to see if they neutralize before they do the work of actually making an antibody. Because making an antibody is a lot of work.
Have you dealt with the monkeys at all?
No. There are 6 primate centers in the U.S. There's not one in Florida. But our animals are at the Wisconsin Primate Center.
What is the process of getting a vaccine approved for distribution?
First, what you have to do when you're in the process is you have to show that your vaccine works on a cellular level in the lab, in a petri dish type thing. And to check if it works there, you would do things like neutralization assays. A lot of vaccines now, their goal is to elicit neutralizing antibodies. So you would, in the lab, check against cells. Is this vaccine actually preventing these cells from getting infected? Are these cells actually producing antibodies against what we want it to produce? That would be the first step. And then once you get that working, you have to move it into an animal model. You typically start with a smaller animal model, like a mouse. Some people like zebrafish, depending on what you're working with, like the nervous system or whatever. And then you typically go to larger animals. Some people go to pigs. We do HIV, so we do non-human primates, rhesus macaques, because it is not ethical to do research on chimpanzees. They are too human-like. And when you get something working in primates, then it is typically, at that point, it's approved for human trials. So then when you get to human trials, the first thing you need to do is checking that the drug is safe in humans and what dose is appropriate.
How do they test for doses?
They give people different doses. I mean, they start at a dosage that's probably similar or equivalent to the animal because they also work out some dosing in animals. So then after they have their dosing and their safety, the next thing is a phase 2 trial. And that's the phase where, like, does this actually work?
What is helping to administer this vaccine through the body?
They're all dissolved into a solution so that it can be administered. RNA is not the most stable of things. This is why these vaccines also have to maintain this cold chain. So there are things in there to help stabilize it, but these things aren't things that cause issues in humans almost all the time. There are some people, as you have heard, that are having allergic reactions to the vaccine, even though it's really rare. I think it's maybe 40 people in the US so far, and they've administered 12 million vaccines. These people are also people who typically seem to have a lot of other allergies. So I think their immune systems are just really overly excited by the wrong things. In a vaccine, you're going to have the protein or a virus or something in there that's carrying the protein so it can actually like deliver. You can't just be like, woo, I'm RNA and I'm just hanging out. But you also are going to need some things in there that are going to be able to keep it stable. Because as I said, RNA is very unstable. DNA is a lot more stable. And things that make sure that everything in there is staying okay. But these aren't things that are toxic to humans. Because I know a lot of times people are like, it says it has aluminum or something. And I'm like, you saw some of the letters of the word, but chemists were really creative when they decided to come up with nomenclature. And it's a completely different thing that's not toxic.
Are there alternative therapeutics to vaccines?
If you say therapeutics, I think drugs, because not all illnesses have vaccines, clearly. Life expectancy would be like 200. HIV doesn't have a vaccine. Hepatitis C doesn't have a vaccine. But both of those viruses, if you're infected, there are drugs. With HIV, we'll stop the virus from progressing. And with hepatitis C, will cure you. So there are drug therapies in lieu of vaccines. I still think vaccines are the way to go. I may be biased as a vaccine researcher.
Do clinical trial participants have to be reflective of the country's population?
It is not a requirement for the participants to reflect the country's population. If you look at the history of a lot of studies that happen in the US, most study participants are Caucasian and male. They only recently started adding females to a lot of these studies. But for the best results and most accurate results, studies should have participants that reflect the population, because even though we are all the same and we are all people, different races and people from different places have genetic differences, and that can affect how drugs and vaccines work. The Moderna trial is actually really impressive because they're very close to what the US population actually looked like.
I've heard the saying, yeah, I'm just going to build my immune system and not take a vaccine. And I wonder, is building your immune system comparable to taking a vaccine?
You can try and strengthen your immune system, right? Your vitamin C, your zinc, exercise, water, eating healthy and all that. Maybe that'll increase your likelihood of having a positive outcome versus the vaccine where your body's already seen the virus. So if you get infected, your body's already ready to attack. Even if you have a strong immune system, if you get infected, you're still waiting those 2 to 3 weeks for your immune system to really kick this virus's butt. So the vaccine causes a quicker response. And what they found in the vaccine is even the few people who are vaccinated who do get infected, they don't have poor outcomes. They may have COVID, but it's really mild. And when we're talking about this virus that's killing 1% of people, I personally really enjoy my life. 1% is really high to be playing with.
Is work a part of your well-being?
For my well-being, maybe like a year and a half or 2 years ago, I started exercising. I think it's a lot. I don't know. I exercise like 5 days a week. And now I actually really enjoy it. So I exercise a lot. I listen to audiobooks a lot. I try not to listen to like serious audiobooks. It's a pandemic. I want to listen to fun, happy chick lit. But I think one big thing that I do for my sanity is I try to never have plans on Sunday. I don't work on Sunday. I don't socialize on Sunday. It's just a me day. And I think that helps regroup and continue on with the week.
That was so validating for me because I too am one who doesn't like to do things on Sundays. And I thought it was kind of selfish or like, oh no, you should still be available to these conversations or to this particular event if it comes up. But I appreciate you saying that because something about it wasn't sitting right with my spirit on Sundays when I was doing things.
You really have to recharge yourself. Even when you're an extrovert and a social person, you need time with yourself to reset.
If you want to connect with her, her contact information is in the show notes. If you want to connect with us, we are at The Research Her on all of the social. We are at theresearchher.com. Please, please, please take the time out to leave a review of the podcast. Short and sweet is all we need. Just show a little bit of love in the podcast reviews. I would really appreciate that. And until next time, I holla.